For aerobic cellular respiration, this occurs when we have oxygen. Okay. The very first step is still going to be glycolysis. This was where we took our sugar molecule and we had to break it down. It was too complex to use on its own, so we have to break it down into something else first. So this was 6 carbons, and we break it down into 2 3-carbon compounds called pyruvate. In the process of making our pyruvate, we are going to generate a net gain of 2 ATP molecules, and we're also going to generate 2 NADH. This is very important when we tally up the total number of ATP produced by aerobic cellular respiration. So we have pyruvate. Pyruvate's going to enter the mitochondria. And the next step, let's go back to purple. The next step is going to be called pyruvate oxidation. So aerobic cellular respiration occurs in 4 distinct stages. The product of each stage goes on to the next one. So entering this stage, we are now inside the mitochondria. We have pyruvate. So still like before, we still have 3 carbons, double bonded oxygen, and another one up here. Okay. Actually, let's move this out a bit. So the general equation for pyruvate oxidation is pyruvate plus NAD plus coenzyme A, and that's going to yield acetyl-CoA, carbon dioxide, and NADH. Important to note here that we have 2 pyruvate molecules, which means this whole thing is going to happen twice. So going back into it, if we look at our products, we see here we have a carbon dioxide being made. So like before, like in ethanol fermentation, this is our pyruvate. We have a COO at the top. This is going to get cleaved. So that gets cut off. And we have a carbon dioxide being produced. So this in the center, this is going to be our acetic acid or acetate. Okay, so that was our CO2. Fantastic. We also have an NADH being made, another electron carrier, proton carrier, and an acetyl-CoA. So coming into here, we have NAD plus again. The plus meaning that it is ready to accept electrons, it is ready to form a bond, and what it's going to do is make a bond with this hydrogen and pull it off. So we pull it off, and at the same time, our coenzyme group is going to come in. So now we have our 2-carbon compound with a coenzyme A group attached to it. This is our acetyl-CoA. So when we pick up that hydrogen, that's going to become our NADH. You'll notice that there's no energy per se being produced here. We don't have any ATP being produced. We do have decarboxylation occurring, the removal of a carbon dioxide group, and we have the formation of NADH. So if we wrote it at the bottom, we'll say we had 2 NADH made during pyruvate oxidation. Okay, so just leave that in screen, and let's use orange. So the next step is the Krebs cycle, or the citric acid cycle. Either one is fine. So when we look at the citric acid cycle, we have acetyl-CoA. Remember, all of the products of one step go into the next. So acetyl-CoA from pyruvate oxidation, it's going to enter the Krebs cycle. This is all still inside the mitochondria. So we have acetyl-CoA, and it's going to combine with something called oxaloacetate. This has 4 carbons. This is going to form citrate or citric acid. And the citric acid is going to be broken down to help yield electron carriers. In this case, per acetyl coenzyme entering, we're going to create 2 CO2. So you know right away we have decarboxylation occurring. 2 CO2, 3 NADH, and 1 FADH2. This is just another type of electron carrier, and 1 ATP. Again, this is per acetyl-CoA, which means this whole process is going to happen twice because we have 2 acetyl coenzyme A groups entering this process. So we go back down to orange. So here we have 2 carbon acetyl-CoA. It's coming into the cycle, and it's going to bond with oxaloacetate. It's going to bond with oxaloacetate, put 2 and 4 together, and we're going to create a 6 carbon compound. 1, 2, 3, 4, 5, 6. This is our citrate. Okay. Now we're at this stage. We need to start looking at what's going to happen. So when we make the citrate, it's not going to be a very stable compound, and it's going to fall apart. At first, it's going to fall apart over here. We're going to lose one of the carbons. We're going to become a 5 carbon compound. Okay, that means this carbon that pops off is going to help make one of our CO2s. To help aid in this process, we do have NAD plus on standby. Plus meaning it's ready to pick up electrons, ready to make a bond, so it's going to pick up a hydrogen and its electrons and help make NADH. Okay, so that's 1 carbon dioxide. We make 2 per acetyl-CoA, so if you guessed it, this 5 carbon compound is going to break down again into a 4 carbon compound. When this happens, we're going to produce CO2, and NAD plus is on standby again, so it's going to help make NADH. Great. Now we have a 4 carbon compound. Oxaloacetate is also a 4 carbon compound, and we want to do this cycle again and again and again because we're going to have a lot more acetyl coenzyme A groups coming every time we metabolize a glucose molecule. So we are going to recycle this. In order to recycle this, we do have to use ADP, meaning diphosphate. So it's going to pick up a phosphate and make an ATP group, which we like. And then on its way back to oxaloacetate, we're going to have FAD, 3E2+. Come pick up hydrogens and make FADH2. And we're going to have one more. So we made 2 NADH. We're going to have one more NAD+. Come by, pick up hydrogen, and make NAD+. So in terms of what we're making, we had 2 ATP over here from glycolysis, 2 NADH, 2 NADH and pyruvate oxidation, Now, for each acetyl coenzyme group, we have 3 NADH, 1 FADH, and 1 ATP. So under here, let's say we made, because this is going to happen twice. So if that's the case, let's say we made 6 NADH and 2 FADH2 and 2 ATP. Okay, pretty good so far. So the last stage over here is called the electron transport chain. Let's use, so this is our electron transport chain, or cytochrome enzyme complex. So at this point, we're still in the mitochondria. In terms of what's going to enter this stage from the previous, it's going to be our electron carriers, our FADH2 and our NADH. So the enzymes that do this in the cytochrome enzyme complex are embedded in the membrane of the mitochondria. More specifically, the mitochondria has two layers of membrane. We have an outer membrane and we have an inner membrane. In the middle, we have a fluid. This is called the Intermembrane Space. Okay, so now our electrons, or sorry, our enzymes that do this are actually embedded in here. So let's draw one like that. Oops. Like that. That. I'll draw a slightly different enzyme over here on the end. Just clean this up a bit. So we said that our electron carriers are going to be entering the electron transport chain. So let's do it like this. And at this point, it should start to look more and more similar to what we talked about in photosynthesis. So if this was just the membrane, this was all just the, it's ugly. This was all just the membrane. The inside fluid, we call this the mitochondrial matrix. Okay, so NADH comes in. And it's going to drop off its electrons that are in between here with the hydrogen. So when it drops it off, we're going to have it turn back into NAD+, and this hydrogen is going to break off and float here in the matrix. Right, so our electrons have entered the enzyme complex, and they're going to go through some redox reactions that we don't cover, and go down to the next enzyme. Same thing, those electrons are going to enter, get energized, and go down. So when we go down this staircase, or when we go down the electron transport chain, we are going to lose energy. When we lose that energy, we can use it to pull hydrogen ions across into the other side. That's exactly what we're going to do. So we lose energy, these hydrogen ions are going to come through, boop, and end up over here in the intermembrane space. So let's say these goes up too, let's say these go up as well. Okay, so now we're having a buildup of hydrogen ions in the intermembrane space. This is the same deal for FADH2. It's going to drop off those electrons, it's going to turn back into FAD+, 2+, and the energy that the electrons provide when they lose it through the electron transport chain will be used to pull these hydrogen ions across. So slowly and slowly, we're going to find that it's going to become more positive over here. It's going to be more positively charged in the intermembrane space than the matrix. So we set up yet again in the intermembrane space an electrochemical gradient. So we have this electrochemical gradient. That means they're going to want to move back down from a place of high concentration to low concentration. So these hydrogen ions come back down through here, and back into the mitochondrial matrix. This last enzyme is going to be the same one that we talked about before. This is ATP synthase. And like before, when they go through ATP synthase, when the hydrogen ions go through ATP synthase, they're going to lose some energy or provide some energy to help stick a phosphate group onto an ADP molecule. So here, now we have ATP being produced. Okay. Now, these electrons that got dropped off by our electron carriers have to go somewhere. If they're not picked up, if they're not removed from here, then the buildup of electrons will actually cause this mechanism to stop working. And then even if you bring more electrons to it, it's not going to go. So to pick up these electrons, this is why we say aerobic respiration requires oxygen. The final product that's going to be produced here when oxygen comes by, when oxygen comes by, is it's going to pick up those electrons. There's a bunch of hydrogen floating in the matrix, and it's going to make a bond and make water. It's going to make H2O. So very, very important end product. So we can see that these electrons that we drop off from our electron carriers help make ATP. The question is, how much? How much ATP do they help us make? So let's tally everything all together. So back at the beginning, glycolysis, we have 2 ATP, go NADH, 2 NADH, and 2 ATP. Over here, from pyruvate oxidation, we didn't make any ATP molecules per se, but we made 2 NADH. Okay, if we look at the Krebs cycle, the citric acid cycle, we made 6 NADH, 2 ATP, and 2 FADH2. Now, in general, we say that 1 NADH makes 3 ATP, 1 FADH2 makes 2 ATP. So if we start to add this up, so we have 4 ATP period being made. So 4 ATP being made there. We have 2 FADH2. So 2 times 2 is going to give us another 4. And now if we look at NADH, there's one little exception. If we look at the NADH that we make during pyruvate oxidation and the citric acid cycle, we have 8 NADH. 8 times 3 is going to give us 24. And now, if we take a look at the NADH in glycolysis, the NADH in glycolysis, they actually undergo another series of reactions and each get converted into FADH2. So in that case, if they were FADH2, they would only make 2 each. So we say that these 2 NADH will make 4 ATP. Right? 2 times 2. So that's 24, 28, 32, 36. So the net yield of ATP in aerobic cellular respiration with oxygen is going to be 36 ATP. All of this is still happening within the matrix of the mitochondria.